Thank you so much to the organizers. Can everybody hear me okay? Great. So I have no financial disclosures. I'm from the National Institute of Health, the National Institute of Neurological Disorders and Stroke. Getting a little bit of feedback. Should I just shut it off? Okay. And here's the outline of my talk today. I will tell you a little bit about my vision for convergence intelligence. I refer to this as a reciprocal interaction between neuromorphic engineering and engineered organoids intelligence to hopefully derive new design principles and learning rules that could lead to energy efficient, smart technologies. Because I'm from the NIH, I obviously have a bias on health technologies, but aspects of what I'm talking about can apply to many adaptive systems that will benefit from power efficiency and adaptiveness. I will end the talk with a rundown of funding opportunities that I think will be of interest to the neuromorphic engineers in the group, as well as outline some meetings I am planning. But first, let me tell you a little bit about myself. I got interested in the brain-inspired engineering space back in 2017 when I was still at Hopkins. I was at a Society for Neuroscience meeting, which is a huge meeting with lots of posters, and I learned about the discovery of a new class of neurons called the phaser cells. These cells are located in the hippocampus and the lateral septum, and as we heard from Terry earlier today, place cells code for location based on firing rate, thanks to the very persistent theta oscillation that is in the rodent while the rodent is locomoting. Well, phaser cells are different in that the phase of the neuron code directly for space. So it has both place-like characteristic and this brand new phase code, which got me inspired. And with my colleagues at Hopkins, we applied for an NSF brain grant, and we secured a grant and developed a novel controller inspired by the hippocampal dynamics. The realization there was that a phaser cell could represent a robot, a group of phaser neurons could represent a group of robots or a swarm, and that the synaptic connections between the neurons is akin to pairwise distances between robots. And so this allowed us to create a dynamic controller that allows the swarm to adapt in a novel dynamic changing environment without any training. And fast forward, I then rotated to the NSF in 2020, and I sort of was very interested in brain-inspired engineering. And that's when I discovered neuromorphic engineering. And I also discovered a really fun topic competition that the NSF's Directorate for Engineering hosts once every 2 years. And the purpose of the topic competition is called the Emerging Frontiers in Research and Innovation. And the purpose of the competition is to solve grand challenges or national needs by bringing multiple fields together. And historically, NSF will run a competition. The top 2 topics is selected and approximately 30 million is invested per topic. And so at that time, I talked to many of you in the audience to gather information on the gaps and needs for neuromorphic engineering. And BRAID, which is brain-inspired dynamics for energy efficient circuits and artificial intelligence, was born out of the realization that learning algorithms and hardware was a gap. And so I was at the NSF for about 2.5 years. And before leaving the NSF, I had the honor of initiating another topic, but this time using organoids as a substrate for biological computing. I left the NSF, but the team of program directors then continued and championed that solicitation, and it's currently on the street, and it's called Began OI. Some of you might be familiar with it. So the selling point here was that there's been many investments in neuromorphic engineering, as you see on this slide, but much of that predated the data revolution, predated deep learning, and most importantly, predated the BRAIN initiative. So the hope of BRAIN is to get neuroscientists as part of the BRAIN initiative to collaborate and interact with engineers in the field. And so here's a summary of the first 2 cohorts of the BRAID awardees, and I'm sure you'll recognize some familiar faces from this room, so congratulations. Now I want to turn and talk about research on brain organoids. As you can see, let's see if I can get my animation to work. Aha. So, oh, sorry guys. So the point this slide is trying to make is that NIH has invested quite a bit in technologies leading up to brain organoids, whereas other agencies have invested in neuromorphic engineering, but the integration of the two has been sparse. So the hope today is that one can use one of the challenges with trying to learn from neuroscience and apply learning rules and objective functions into the algorithms you're interested in designing is that it's very hard to actually understand what's happening in an awake behaving rodent. So the thought here is, well, if we could use organoids, not just simply as a new substrate for compute, but also as a complementary model system where you reduce the minimal set of what's needed to then learn the task. Can you imagine we actually would have a chance at being able to learn from these new experiments that were designed with neuromorphic algorithms and hardware in mind? So this is the gist of what I view as convergence intelligence. And I got a minute back, okay. So I'm just going to show you a couple of examples out there to bolster confidence that cultured neurons could in fact learn. My movie. This is work that was published a couple of years ago, in which you could see that both mouse and human cortical cells is able to learn the game of Pong after 5 minutes of gameplay, while the controls was not. The controls was simply buffer and cultured neurons that received either random inputs or no inputs. And this study shows the importance of feedback for learning. The information about the location of the ball was delivered through these electrodes up in the sensory region of the microelectrode array, while the information to drive the paddle movement was delivered through the motor area regions. And the next example is work out of the Pasture Lab, in which they figured out how to take a human cortical spheroid and a human spinal spheroid, as well as a human skeletal spheroid, attach them together in what's called an assembloid. And you can see here from the movie that when you stimulate the cortical part of the assembloid, the musculoskeletal part contracts. And this is really exciting to me because by extraction, if you could teach cultured neurons how to play Pong, I would argue that you could teach this assembloid how to play Pong. The inputs could be the stimulation vector, whether it's the frequency or the amplitude, and the output could be the actual contractile level in response to the stimulation. Although I suspect that was not the intent of Sergey's experiment when he devised the assembloid. Now, some of you might be wondering, well, it's probably really hard to measure microelectroarrays of organoids if the arrays are two-dimensional, and you would be correct. So there are people developing flexible electrodes that you can throw into the organoids, and it will grow with the organoid over time, allowing you to characterize it with more precision. This is work from Harvard out of Jalou's lab. And and then this other example is from John Rogers' lab, where he has developed a three-dimensional multifunctional neural interface for cortical spheroid and engineered assembloids. And you can see there are at least 4 different kinds of modalities in this device. And I want to change gears a little bit and talk about something a little different that's done here by Duigu Kuzum. And she's actually taken a human organoid and successfully intercalated that into a mouse model into the retrosplenial cortex, which is a vascularization-rich area, and showed functional connectivity. And what you're looking at here are see-through graphene electrodes in the middle panel, and the red traces belong to the organoid. Sorry, the red squares and traces to the far left belongs to the organoids. And this is an important study because it shows us how combining stem cells and neural recording technologies is promising for modeling diseases, for personalizing treatment, and for the organoid's potential to restore lost function. And this last example I want to show is from UC Irvine, in which they're trying to use engineered neuroglia cultures for neurorestoration after stroke. And the idea here is, in order to even do this, you have to be able to read out EEGs from the patient. In this case, they're looking at finger movements. But the first thing they did was they first demonstrated that their culture prep can spontaneously spike. And after they demonstrated that, they then showed that they can in fact train it by using 600 pulses applied to the most 2 sensitive electrodes in the MEA patch. And they show that training stimulation affects dramatically the culture, as you can see here. So I don't have time to go into much details, but I hope this gives you a sense of some promising happenings. Can't seem to get my slide to advance. Phew. Okay. So I hope I've shown you that convergence intelligence is ripe for exploration, that there is an opportunity here on organoid-informed, nervous system-inspired neuromorphic technologies. You all know that neuromorphic engineering is showing promise beyond simply power efficiency. I mean, power efficiency is good. Here, I see organoids and assembloids are emerging as promising design platforms, both as a new substrate for computation and also as a simplified model organism to help us learn what biology is doing. Yet organoids and assembloids and neuromorphic engineering and neurotech are all on separate research tracks. So it is my personal opinion that deliberate efforts across government, industry, and research community is poised to accelerate the promise of convergence intelligence. And why stop at just a few model systems? The nice things about organoids is that you can just get the skin cell, you don't have to sacrifice the animal and then reprogram the skin cells. The nice thing about neuromorphic is that it's very power efficient, it's adaptive. So organoids can be gotten from the patient while neuromorphic technology is extremely efficient. So this just feels like a collision that should happen to create the next generation of super efficient, powerful, and personalized health technologies in which to minimize things like foreign body responses. So I see that I'm out of time, so I want to just get to a couple of funding opportunities. This is the Begin OI. It has a due date of December 24th, and letters of intent are required. The next Emerging Frontiers in Research and Innovation call is going to probably happen this fall. If anyone is interested in pitching a topic, I'm happy to talk to you. These are 3 funding opportunities on the street. Brain behavior, quantification, and synchronization seeks to monitor humans and animals for extended periods of time. This is where neuromorphic has an advantage because power efficiency is at a premium. Well, the complementary program seeks to develop novel approach methodologies. Organoids is one such system, and the Brain Initiative, of course, is they're always interested in high risk, high reward neurotechnologies, whether it's neuromorphic or not. CRCNS is another program that supports both neuromorphic computing and organoids research. And if Robinson Pino is in the room, he is the Department of Energy representative, and he should be here at this meeting. And so finally, I want to end on the 4 meetings of interest that I'll be organizing the rest of this year. If anyone's coming to the Brain Initiative meeting, sorry, it's now called the Brain Conference, we will have a special session on neuro and AI on June 18th. Dewey, you and I are hosting a symposium at SFN in Chicago on advancing organoids. And I'm in the process of co-organizing 2 other neuromorphic neurotech workshop. Hopefully it will take place this fall. So if you're interested in creating technologies for the health domain, please find me. I'll be here all week. I'm even going to be here on Friday for the tutorial. So thank you. Any questions? Any questions for the speaker?
Please, only to see. I have a question about the brain research to the innovative neurotechnologies. Are these technologies for human or can be for animals?
Yes.